right, so let's talk about chapter 3, cells. Yeah, I am not trying to make anatomy and physiology any more complex than it already is. So if you're asking yourself, do I really need to understand cells and these microscopic things that I will not ever see outside of this class? The answer is yes. You have the tools to affect a patient down to a cellular level. I mean, think about that. You have the power to affect a patient down to a cellular level. And it's as simple as oxygen. Do I give my patient oxygen? Do I not? It affects at a cellular level. I mean, just kind of give that a second. Give that a second. So yes, we have to understand cells in anatomy and physiology. And the way I want you to approach learning about cells, because it seems just so ambiguous and microscopic and you can't touch it, and I want you to think of it this way, okay? I want you to think of the human body, right? The human body has all of these components that are working together. We have cells that make tissues, we have tissues that make organs, we have organs that make organ systems, and organ systems make up the human body. And they all are working together, right? We're all working for homeostasis, optimal performance. And cells are these individual microscopic entities, right? These things that all have components in them that allow them to function in their role of the human body. So, for example, I have a cell that has organelles in them that are functioning together. Those organelles are functioning together so that we can make some ATP and we can make some proteins and we've got lysosomes that are going to help with digesting any foreign bodies or garbage, and they're going to digest things. And I want you to think about, well, that kind of all sounds familiar, like the human body. The human body has a system that will digest things. The human body has a system that will protect against invaders. The human body has a system that will function in communicating information back and forth. So cells are these microscopic units that function like the human body. And when you kind of look at it that way, it's kind of cool, right? So let's dive in. All right, here are a bunch of cells, and I think this slide is going to really help bring in what I like to say and probably say quite a few times while we work together, is in that form follows function. Let's look at this neuron, for example. This neuron here, it's from the brain, as the slide says, And we know that the nervous system is responsible for communicating a lot of things throughout the body. And they need to travel and send that information across the body. And just looking at this cell, just the branching and then the branches off of that in every direction, and the form of this cell itself is allowing it to function optimally. So form follows function. And I want you, as you go through anatomy and physiology, bring it back down to how does this cell, how does this tissue, how does this organ, how does this organ system, how is its form allowing it to function optimally? Because if we can understand that, we can understand that when it's not working, and we can understand why we have the signs and symptoms we do. And we can then anticipate the direction that our patient's going. And therefore, we can be maybe a step ahead or two ahead to give them optimal care. So as we talk about cells, first thing that we have to recognize is that cells are the building block of life. They're the building block of life, okay? There is a theory known as cell theory, and it has 4 components that we should take note of. And it's this, that again, all cells are the building block of plants and animals. They're the building block. We need cells to build. Cells are the smallest functioning unit of life. Now you're going to hear me say the nephron is the smallest functioning unit of the urinary system. You're going to hear me say functional unit, smallest functional unit of fill in the blank for nearly every system that we study. The cell is the smallest functioning unit of life. Okay? Cells are produced by the division of a pre-existing cell. Okay, what does that mean? Well, that means that we have a cell that will divide and replicate itself to make other new cells. The idea that I have a cell that can divide, well, how does it know what it needs to divide into? That's a little deep. However, I want you to understand that there is an origin cell. An origin cell being the stem cell, which is an unspecialized cell. It's unspecialized in that it doesn't know who it's going to be. It doesn't, it just knows that where it's at, it needs to divide and differentiate into, let's say, this nerve, this neuron, okay? In order for me to have more cells, I have to divide and be produced by a pre-existing cell. Okay? And the part that's going to really bring home the fact that cells function like the human body is this last one. Each cell maintains homeostasis. Each cell maintains homeostasis. So every single cell is working to maintain homeostasis. Okay? All right. Anything that ends in logy, ology, is the study of. Okay? And if I told you that cyto is cell, then you would be able to tell me that cytology is the study of cells. So one of the other big things about anatomy and physiology, if you haven't picked up already, is that you're learning a new language. And part of it is breaking it up and understanding what each of the parts of the words are so that you can, maybe you don't know what the answer choices are saying, but if you're able to break down the terminology, you might be able to deduce the right answer. All right. Let's talk about the cell properties. All right, so talking about some of the components or properties of the cell, the first thing that we'll start with is the plasma membrane. Plasma membrane is that outer surface here, outer surface that is essentially the boundary that keeps the organelles on the inside of the cell and keeps things that don't belong in the cell out. Now, the other thing that the cell is going to have is a cytoplasm. And it is going to, it's a fluid that is going to keep the organelles surrounded, and there's also going to be a fluid on the outside of the cell known as the extracellular fluid or interstitial fluid, which is in between the cells. Now, speaking again about the plasma membrane, this membrane is going to do some things, right? Naturally, you can deduce that it's a barrier, it's protecting, right? It is going to also offer an opportunity to exchange material with the environment. It's going to be responsive to the environment, so it's sensitive to the environment. It's going to have receptors that are going to help interpret the environment so that the cell knows, if for example, if the level of oxygen outside the cell is too high, then the cell might make ready to take in some oxygen. And the other thing that the plasma membrane is going to do is it's going to produce structural support for the cell itself. So looking at the plasma membrane, let's look at some of its components. So we see here that there is these red little circular objects, and they seem to be attached to these threads. And we've got some more threads in the opposite direction, and then these red circle objects. So one of the features of the plasma membrane of our cells is that they contain a phospholipid bilayer. Okay, and so it's a bilayer, so this is one layer here, and this is the other layer, and you can see that they are in opposing directions, right? And phospholipid, phospho is, a phosphate group. Lipid is a fat. And so on this surface here, we have these circular objects that are soluble in water, meaning they like water. Hydrophilic. They like water. So water, great. It can pass through this membrane, right? Maybe. Because the other component of this layer is a lipid layer. And water and lipids, they don't go well, right? They don't like to mix, okay? So those those lipids, they are hydrophobic. They fear that water. Ugh. So I like to remember philic is I like it a lot, and phobic is I fear it. Okay? And so this means that this membrane may or may not permit things across, which is good. And we define that as a semi-permeable or selectively permeable membrane. Okay? So if I asked you what is the plasma membrane of most of our cells in our body, are they semi-permeable? Are they freely permeable? Or are they impermeable? You would naturally say that they are semi or selectively permeable. And I trust that you are intelligent, and you can deduce that if I said the membrane was freely permeable, that, well, everybody got through, right? no discrepancy. Everybody got in or out. And an impermeable membrane would be a membrane where nothing was in or out. Okay? Lockdown. Okay? So, talking about the structure, the fact that the plasma membrane allows for structure, I want you to focus on these cholesterols, these yellow cholesterols. They are giving the plasma membrane structure. They stiffen the membrane, okay? So not all cholesterol is bad. Some cholesterol we need. And one of the areas where cholesterol is functional is in our plasma membrane because it helps to provide a stiff, rigid rigidity to our membrane. And then if you look, we've got these big old blue proteins. These big old blue proteins. So I want you to carry with you for now and forever that proteins are large. I mean, look at the size of these proteins here. They're large. Okay? I want you to hold on to that as well. So, talking about membrane proteins, there are a variety of membrane proteins that all have different functions. So, there is a table on 63 that I would, if you're a table kind of person, go ahead and reference that. But it's all kind of in the language. So if I said that I was a channel protein, for example, this one here in the middle is a channel protein. Well, just think of a channel. A channel is a passageway, right? This protein is a protein channel acting as a passageway for material in and out. Okay? If I'm a protein gated channel, well, I'm still a channel I can pass through, but I have a gate. So I might need the password, or maybe I need the key in order to get through. Fair? Okay. One thing I want you to take note of is that an enzyme, which we'll talk about again later, an enzyme is a protein that is going to encourage a chemical reaction without using as much energy. So we'll talk about that again, but I just want you to, now you've heard the word enzyme, so when it comes up again, you should kind of be like, she said that before. Something about a reaction, okay? And that it's a protein. All right? Okay, so carbohydrates. Carbohydrates are going to be within the membrane as well. So carbohydrate chains up here and up here. Carbohydrate chains are going to help with lubricating. They're going to help with adhering cells to one another. And they are also going to be part of the recognition system. So they also, I mean, as they're kind of sticking out here like antennas, it's sort of like having a flag that's like, hey, I am this kind of cell. So the body knows what it is. So 3 things those carbohydrates are doing, right? They are lubricating, they are adhering, and they are communicating like a flag. This is the country of the United States. This is what I am, right? All right, great. So let's talk about diffusion. Diffusion is, we've got to talk about the idea that diffusion is passive. What does that mean? It means that it doesn't require energy. It doesn't require energy. So this is a process that doesn't require energy. If I was a process that required energy, I would be active. Active means I need energy to do whatever it is my function is, okay? So diffusion is a passive process, okay? And there are different types of diffusion, and we're going to talk about just a simple diffusion here. You can easily do this in a glass at home. You can, we will do this in the lab. We'll talk about diffusion. This is a very important physiological process, and believe it or not, it is how our treatment for our patients is, for the most part, how it's going to make them either better or worse is through this idea of diffusion. Okay, and so the first thing that we have to talk about in diffusion is the fact that we have a concentration gradient. Concentration gradient is, I take a bunch of particles, and I put them all together in a small space, okay? Small space. And my concentration in this area of these particles, let's say they are oxygen particles, is really high because I packed them all in here, and it's really, they're all packed, it's really tight, okay? Concentration of my oxygen in this area is really high. Always compared to something, okay? Compared to this entire room. I put a lot of oxygen. I grabbed it and I tucked it into this little area that I put in my hand, and I put all the oxygen that I could. And then in the rest of the room, I've got some oxygen. I do have some, but I also have some nitrogen, and I have some carbon dioxide. And so I have created a concentration gradient because the container that I have, let's say in my hand, that is holding only oxygen, that oxygen level, the number of oxygen atoms that I have is high compared to the outside immediate environment. Let's say I've got 50 oxygen atoms in my little container here. I've got 50, but there's only 25 oxygen atoms around outside this container. So the concentration gradient by nature is going to go from high to low. by nature. Nature just wants to balance and equal out. So what happens? I open up this container, and does the oxygen stay in the container? Or does it move into the environment? Well, the answer is that it's going to leave the container and move into the environment. It's going from a concentration that is really high to a concentration that is lower in the environment. The same here in this diagram. We can put a sugar cube, we could put a salt cube in a beaker of water. And what is going to happen is this concentrated block into water is going to move from the area of high concentration to an area of low concentration, as you see in number 4, where it's just sort of spread all throughout. You can do this with a glass at home and some ink. You drop a couple of drops of ink and you can watch as it spreads throughout the container from a high concentration to a low concentration. This is the process of diffusion. Okay? High concentration to low concentration. And it is passive. It is a natural process in nature. Nature just prefers equilibrium. Okay? So, now, why am I going to move across a membrane? Because we talked about membranes and now we're talking about diffusion. And so why would, say, sodium move across a membrane? Okay, let me paint a picture. Okay, you've got a cell. And inside the cell, you have maybe 5 sodium atoms. On the outside of that cell, in the environment, you have 100 sodium atoms. So, now that we have talked about some of the rules of concentration gradient and talking about nature and wanting to find equilibrium, do you think the sodium is going to, that's on the outside of the cell, do you think it's going to remain on the outside of the cell, or do you think that it's going to move inside the cell? It's going to move from the area of high concentration, so the area that has 100, and it's going to move to the area that has less concentration, or inside the cell where there are only 5. And it's going to do this until it gets to equilibrium. It is nature. Okay? So the fact that our membranes are semi-permeable means that some things are going to be able to diffuse across the membrane, and some are not going to be able to cross the membrane. And some may be able to cross the membrane, but maybe with a little bit of help. And that's where we get our different proteins that maybe help facilitate. Like facilitated diffusion, for example, is still a passive process. It's still passive, but it needs a little help from the channel, okay? It's facilitated. I facilitate your education in anatomy and physiology. I guide the way. Same thing with facilitated diffusion. I have a channel that is going to help a gas or a protein or whatever across the membrane, to diffuse across the membrane. We also have this special type of movement of diffusion, and it's called osmosis. Osmosis is actually the movement of water. So you need to be able to understand the difference of diffusion, and you have to understand the definition of osmosis. So with osmosis, we have to talk about solutions. Solutions like this image. Initially we have a beaker filled with a solvent. Typically in the body, the solvent is water. We are 75% water, 60 to 75% water, and our solvent is water. So in this beaker, let's say it's water. You then are going to put this, let's say, sugar cube. This sugar cube is a solute. It is the material that will diffuse. It's in a smaller quantity. It's not the fluid. It's in this case it's sugar. Okay? And as we go through putting the sugar cube in to the beakers, we mix them up and we get the diffusion. The sugar diffuses across all of the solvent, and now we end up with a solution. And it's nice and mixed, like in number 4. So solutes are going to drive water. Let me tell you what I mean. Water is going to go from an area of high concentration to low concentration, just like salt did, just like sugar does in diffusion. However, water is going to follow the solute concentration and work to dilute it. Okay, so let's wrap our head around it. So I've got this cell that has very little water in it. It has a lot of sodium in it compared to the outside environment. Remember, always compared to the outside environment. On the outside, I have water, a fair amount, nothing more than what I should have, but I have more water compared if I was looking on the inside of the cell, though. And so, although there is a difference in the concentration of water between the two spaces, It's actually the fact that I have all of this sodium on the inside of the cell that water is going to go and diffuse across the membrane through the term osmosis in order to dilute the concentration of the salt. Take a second and make sure you get that, okay? Because it's the movement of water, yes, from a high concentration to a low concentration. That is osmosis. But the driving force is the concentration of the solute. And essentially, if you think of it as I need to dilute this solution, that is osmosis. Okay? So the other part that we need to talk about in osmosis is osmotic pressure. So when we talk about osmosis and we consider what is osmotic pressure, I think this slide does a nice job. It's on page 66 of your book. And so this is talking, we've got this container and we've got a mixture of water and solute. So we've got a solution. Now, if you look at number 1, the content of side A has more water molecules than side B. And you can actually count these out and count the solutes out, and they are different. Okay? And so this idea of osmotic pressure is that the force of water across a membrane produces a pressure. Okay? So we've got this semi-permeable membrane between these two, A and B. And once, again, through osmosis, the movement of water, right? And the movement of water is driven by the amount of solute. Great. Okay, perfect. Side A then is going to proceed to side B in efforts of diluting the solute, and therefore we'll have a decrease on side A and a volume increase on side B. I can measure this force of water across this membrane by applying pressure, applying a force as that water diffuses or through osmosis moves across that membrane. And I can measure it to see the osmotic pressure, the movement, the force of that water across that membrane into the other side. I can measure that, okay? If I apply force to side B, I can generate a hydrostatic pressure which will help equal out the two sides. Okay? This is important because this is how Filtration happens in the kidneys. This is how resources cross, like water crosses membranes to control blood pressure. So this is important to understand. So again, we've got osmotic pressure, which is the force of water in a solution. It's that movement in a solution across the membrane in order to equalize, or not equalize, but to dilute a solute. And then we have hydrostatic pressure, which is that pressure that I can put down against I guess, how would I say, against an opposing pressure. So hydrostatic would be against an opposing pressure. And that hydrostatic pressure is going to oppose the osmotic pressure so that it becomes equalized across the semi-permeable membrane. Make sure to really look at page 66 and break that down and understand osmotic versus hydrostatic. And let me know if you're struggling with that concept. Because again, it is the driving force of blood pressure, of filtration in the kidneys. So this is a physiological process you want to make sure that you can kind of wrap your head around and kind of talk it out. And make sure you understand. So the other part of osmosis is the idea that we have fluids in our toolbox that we can give that may change the amount of water a cell has within. Okay? So tonic is tension. And iso is equal. So equal tension. Okay? I'm thinking tug of war. Okay? And I'm thinking that equal tension is both sides are pulling the exact same tension and we're going nowhere. Follow? Great. Isotonic solutions are doing just that. Water in isotonic solutions, solutes in isotonic solutions are playing tug of war. Both sides are equal. Tension is equal. So, are they exchanging resources, or is this volume staying in the container I put it in? Yeah, it's staying in the container I put it in. Okay? It's not crossing into the cell. We're not exchanging anything. It's staying in the container. If I am hypotonic, so let's say this hand, my left hand is hypotonic, okay? and I'm playing tug of war, I'm going to go towards my right hand. My right hand is going to have tension. My left hand is just going to go along for the ride. So essentially, my right hand is winning the tug of war. Okay? Now, what does that mean in a cell? I'm glad you asked. In a cell, it means that I have more solute inside my cell compared to the outside. Always compare. Okay. So I then have more water on the outside of my cell compared to the inside. And if I'm hypotonic, am I going to be pulled in to the cell or drawn out of the cell? If I'm looking at water, I'm going to be drawn into the cell. I'm going to be pulled in because I have a lot of solute on the inside compared to the outside environment. And so I'm going to go ahead and go and dilute the inside of the cell. Okay? And I'm going to do this until I can't do this anymore. Either I don't have any more solution, hypotonic solution, or I'm a cell and I simply have no room and I rupture, also known as lysing. Also known as lysing. Okay. If I have ISO, which means nothing is happening and it's just staying in the container, and I have hypo, where I'm being pulled into the cell to dilute the solute concentration, I must have a hypertonic solution, and I do. In my hypertonic solution, I have more solute in the solution than I do inside the cell. So I have more solute outside the cell compared to the inside of the cell. So the water from inside the cell is going to remain inside the cell, or is it going to be pulled out of the cell and into the outside environment? Okay, good. It's going to be pulled out into the environment because I have a lot of solute in this solution that I've given. So I'm pulling water out of the cell and into the environment to dilute my hypertonic solution. Good. Now, I'm going to do this until I don't have any more fluid, I'm balanced out, or until my cell shrivels and crenates and dies. So, let me ask you this. I said from the beginning that you have the power to affect your patients at a cellular level with just oxygen. You also have the power to affect your patients at a cellular level with fluid. I mean, is it really that serious? It can be. So let me run this by you. You are in your paramedic program and you worked on your IV skills. You've owned it. You're great. You've done like 50 of them. It's just no problem. No problem. And you get out into your clinicals and you're doing your IVs. You're having a good day. You're not missing. And you start a line and you are told to give a hypertonic solution in the form of, let's say, D50. Okay. And your line seems good. All is well. No complaints from the patient. And you administer the D50, your hypertonic solution. But you were having such a good day that, well, you missed one. It happens. And you didn't check to make sure the line was still good. And you pushed that D50 anyway. And you didn't realize that it was no longer in the vein. Okay. It's going to be fine. It's going to be fine. I mean, it really is going to be fine. No, it really is going to be fine. However, what is going to happen to the cells in that surrounding tissue now that they've been exposed to a hypertonic solution that is not in the vein and is in the tissue around the vein? What is going to happen? So, is nothing going to happen? Like in isotonic, nothing happens. It just stays in the container and there's no problem. Is it going to lyse or rupture like in hypotonic? Or is it going to shrivel and crenate like in hypertonic? Okay, well, it is a hypertonic solution. So, okay, you're right. Yeah. It's going to crenate. Okay, so what? Well, if crenate means that everything, the water from inside the cell is going to exit the cell and the cell is going to shrivel and it's going to die. So what happens to that tissue that has hypertonic solution all around now? Potentially necrosis, right? Okay, so it will be fine. It would certainly be something to report, right? And make sure that we manage that right. But you get the idea, right? So you can change a patient's whole health, body, whatever, at a cellular level. And that is my favorite way of talking about how even fluids, we think, oh, it's no big deal, it's just fluids. No, it's a big deal. It's a medication, and we need to make sure it's appropriate for the patient, for that time, for that condition, etc. Okay, and you'll learn all of that. But okay, I'm gonna show you this picture here, and then I'm going to finish out this chapter. Diffusion and osmosis is really some heavy material, so I think we nailed it. If there are questions, specific questions, make sure to reach out. Don't go down the Google journey. Let me help facilitate and get you really good resources if you're struggling with anything. But I need you to be solid in the concept of diffusion, the concept of hydrostatic and osmotic pressures, and the concept of osmosis. Okay, you need to be just like zinging it out like it's natural. Okay, and if you're not, and if you've got questions, you need to reach out to me right away. This is fast paced, so reach out to me right away. Okay. All right, so I am going to move over to the next section here, where we talk about the rest of how things move across the membranes, and we'll get into the organelles of the cell as well. Okay. All right, so remember I said that there was that facilitated diffusion that is still diffusion. It's still passive, but it's that idea where I'm going to help you move across a membrane. So I'm a channel that helps you move across the membrane. So I like to think of it as a channel that will open a little bit, let you in, and then I'm going to let you out. I'm going to let you in, and then I'm going to let you out. The idea is that this channel is only allowing, in this image, just glucose, and it's literally just facilitating. It's like kind of squishing it all the way across the membrane. Facilitated passive process, no ATP, but it is different than simple diffusion in that the rate of the transportation across the membrane is not indefinite. It is restricted to how many channels, how many protein channels there are, facilitated protein channels there are, and the fact that it's only doing one at a time, right? Or in this instance, doing one at a time. So it is not indefinite, right? So the rate is going to be limited, right? The rate of diffusion is limited by these channels, okay? Active transport, again, requiring ATP, requiring energy. Things that you can attribute to active is our ion pumps. Okay, ion pumps. Ion pumps like sodium, potassium, calcium, magnesium. Those are ions, right? Remember from chapter 2, or you'll remember it once you get there, depending on how you're looking at these lectures. But these are ions. They are charged and they require carrier proteins to transport them, and those carrier proteins require energy in order to transport them. There is a special kind of ion pump that we need to talk about, and that is the exchange pump. And the exchange pump is that the ion is being switched. So a great ion pump in the body is the sodium-potassium exchange pump. Sodium is going to be pumped out of the cell, while potassium is pumped in to the cell. Okay? Now, it's not a 1-for-1. It's 3 sodium out and 2 potassium in. And it's a ion pump requiring energy because sodium is going against the concentration gradient. So it's going from an area of less sodium to an area of more sodium. So I'm needing to really get that energy to move it out of there, because it's naturally, it doesn't want to do that. That's against nature, right? So, and potassium is the same. Potassium is going from low concentrations on the outside of the cell, and it's being forced into the cell where there is more potassium, and so it is requiring energy to do this. And the sodium-potassium exchange pump is a very very big concept, a very big transport system in the body. So here you have the sodium-potassium pump, like I said, 3 sodium out, 2 potassium in. And we're going to talk about, and I'm going to take an opportunity here just to explain and sort of set you up for later on in the material. So when I compare the outside environment to the inside environment of my cell, let's say I'm looking at a nerve cell. Let's say I'm looking at a nerve cell. The inside of my cell, inside my cytoplasm, I have a lot of proteins. Remember, proteins are pretty big, right? And they have a charge. And then I've got some other things here, like I've got some potassium in here. They're kind of small, especially compared to my proteins. So they don't carry as much weight as far as charge goes. And proteins tend to have a negative charge, negative energy. So that means when I look at the inside of my cell compared to the outside of my cell, the inside is negatively charged. Okay? It likes to be there, especially if I'm a nerve. I like to be there. I like to have a negative energy inside my cell because I'm excited. I'm excitable. In order to do my job, I am ready to go. I'm on my toes. I'm excitable. Okay. And so when I am at rest and I'm a nerve, the inside of my cell has a charge of -70. That's just, what? And I know, I'm introducing it to you now so that you can kind of wrap your head around the fact that the inside of the cell has a charge of -70, and it just doesn't sound like that makes a whole lot of sense. But we'll build on it. So hang on to that information, okay? Your sodium-potassium exchange pump, very important, especially when you get into pharmacology. So know it, feel good about it, okay? All right. We're going to talk about how things get into the cell and how things get out of the cell. They get into the cell through endocytosis. Endocytosis is the fact that the receptors on the cell are going to recognize some messengers, and they're going to go, I need that in my cell. I need that inside my cell. And so they're going to endocytose by kind of wrapping and grabbing and closing. And what they do is they create this little cavity. I just saw that just made some hearts. So there you go. It's going to make this cavity, we're going to call it a vesicle. And so now on the inside of my vesicle, I've got whatever material was out there that I needed to bring in to my cell, okay? Remember those lysosomes I talked about? Those lysosomes, they are the digestive organelles. Well, we're going to get some lysosomes and we're going to open up that vesicle and we're going to remove the particles that were on the outside so that they're on the inside, okay? So we're going to digest, we're going to open up that vesicle, we're going to dump it out. It's kind of like, I think of it as Amazon dropped a box at my front door. I love Amazon, okay? Amazon dropped a box at my front door. It was brought into my house. It was all closed up and packaged. My lysosome is my box cutter, my box opener, right? Because, yep, I have one of those because I love Amazon that much. And so now I am using my box cutter to open up my box. So I'm a lysosome and I'm opening it up, right? And then I'm going to take out the inside, whatever it is that, whatever present I got. Yay. Okay, so once I do that, now my vesicle is empty, and or let's say I opened up that box and I had a bunch of smaller boxes that needed to go out to the recycling bin, so I put them all in that big box, packaged that up, and now I need to take it out of the house. So in order to get it out of the house, out of the cell, I'm going to use exocytosis. So I'm going to take this vesicle, this box full of all the other little small boxes, and I'm going to go ahead, bring it up to the plasma membrane. It's going to become, it's going to merge with the plasma membrane, and then it'll open up to the environment, and I drop my recycling in my recycling bin. Hopefully that makes some sense. Again, if there's any questions, let me know. Another idea of endocytosis is a term that you're going to hear a lot of. It's phagocytosis. Phagocytosis. That is a cell's ability to eat. To eat. And basically what it does is it kind of the same thing as endocytosis, just in general, just talking about endocytosis, is it's going to come up and it's going to eat something from the outside and bring it in. So I want you, in this instance, phagocytosis, I want you to think of an invader, a bacteria, virus, whatever. And your immune cells, I want you to just think that they come up, your white blood cells, they come up and they grab, they kind of extend their cytoplasm to kind of make these little hands, and they bring it in, and they eat phagocytosis. Okay, all right. And here is an image of phagocytosis. So the cell just kind of using the pseudo-podium, so false feet, and it kind of extends, and then it just grabs that bacteria, brings it in. And then, of course, your lysosomes, they're going to digest everything, right? And break it all down so that you can get rid of the trash, right? All right. If you're a table person, there's a comparison of diffusion versus osmosis, and active versus passive membrane transport on page 74 if you're a table kind of person. Okay. All right, so we're going to talk about some of the features of the cell. We talked about the cytoplasm. Also, you can call it the cytosol. It's the intercellular fluid. And it's going to have things like ions, proteins. It's going to have amino acids, carbohydrates, things like that inside. So the organelles are little organs. That's exactly what they mean, little organs. And so when we look at the little organs, the organelles that are within a membrane, enclosed in a membrane, we're talking about the nucleus, the mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes. And when we're talking about non-membranous organelles, we're talking about things like the microvilli, the cytoskeleton, the cilia, flagella, ribosomes. So we're just going to talk, I'm going to talk about what I think you should know. I don't think you should know every little detail about every one of these things, okay? So go along with me and know what I'm talking about, okay? So the cytoskeleton is just that. It's the skeleton. It is 2x4s that hold up the house, that give it the frame. It's the bones that hold the human body together. Okay. And then we're going to have microvilli. Microvilli are extensions on the cell, or projections on the cell, that I like to describe them as like fingers. Okay, they're kind of stumpy. They're fat, but advantageous, because I mean, think about it. Like, I've got these little projections on this cell, right? And it's not flat, because if it was flat, then I would only have this surface area, right? I would only have this surface area. But if I have these projections, I have these fingers, look, I've got all this surface area, a 360 surface area here, 360 surface area here and here. So I think of them as fingers, okay? Microvilli. And so you're going to find those in the digestive tract. You're going to find them in the kidneys. And we'll talk about more of that as we go through. Cilia is hair. Like, I think of them as hair-like structures, okay? And they're slender. They are going to help create wave movements, things like helping to move things, like an irritant that is in the respiratory tract, for example. They're going to create wave-like movements so that we can get that irritant back to the back of the throat, that cilia, the hair-like structures. So they're going to kind of capture things and then just kind of create movements. We also see this in the reproductive system, where we can kind of move the cells of the reproductive system in the direction they need to be going. Flagella is just this long whipping tail that goes kind of circular. In the human body, the sperm cell is the only cell that has flagella. All right. Ribosomes are going to make proteins. Okay. Proteins are essential for life. They're the building blocks of everything. Protein. So ribosomes are going to make protein. There are free ribosomes and there are fixed ribosomes. Fixed ribosomes are going to be found on the endoplasmic reticulum, and free ribosomes are going to be just within the cytosol, just functioning within the cytosol. We're going to talk about the endoplasmic reticulum. There are 4 things you need to know about the endoplasmic reticulum. 4 things, okay? Endoplasmic reticulum makes proteins, carbohydrates, and lipids. So it makes. So it's a manufacturer facility. It's making carbohydrates, lipids, and proteins. Okay. Since I made it there, I need to store it there, because where else am I going to store it? So I'm a manufacturer facility that can also store. Okay. I'm also going to be a highway system within my manufacturing facility that I can transport things from one place, from one spot of the cell to the other, to another part of the cell. So I'm a highway system that I can transport things. The last thing most people forget, but is actually kind of unique and kind of cool, is that the endoplasmic reticulum is going to absorb drugs and toxins and neutralize them. So drugs and toxins are going to be neutralized by the endoplasmic reticulum. Neutralizing meaning you are no longer an effective drug or toxin, right? I've neutralized you. Neutralize the enemy, right? Okay. The Golgi apparatus is these flattened discs. And there's about 5 or 7 or so. They're flattened. And they are going to package things like hormones and enzymes to be released from the cell. So think of the Golgi apparatus as my Amazon factory who is packaging up my boxes. And specifically, they're going to be packaging up things like enzymes, again, which are proteins that help chemical reactions move forward with less energy. Great, fantastic. But also hormones and enzymes. All right. And they are also going to, the Golgi apparatus is also going to help repair, or not repair, but renew is what the book says. Renew the plasma membrane. And remember those lysosomes? Yep, the ones that digest. Good. Okay. Those lysosomes are going to be packaged by the Golgi apparatus. Okay. And then we talked about the lysosomes. You are aware the mitochondria is the powerhouse of the cell. It is going to make our ATP. Great, we're good there. The nucleus is the, how do I say, the nucleus is command center. Okay, that's where the information is. That's the data center. We're going to have our genetic information is going to be in our nucleus. The nucleus is going to be surrounded by a nuclear envelope. Okay. And the DNA or the RNA, depending on what kind of organism, right? The information, the genetic information in the nucleus is stored in humans as chromosomes. Okay? Chromosomes. So there are, and I need you to pay attention, there are 46 chromosomes. 46. chromosome in the human body, in the cells. There are 23 pair of chromosomes in the human body. So really watch. Is the question asking you about how many chromosomes are in the human body? 46. Or is it asking you how many pairs of chromosome are in the human body? 23. Of the pair, the 23, one pair are going to designate your genetic sex. Okay? That is pair 23. Okay? The 22 other are your autosomal chromosome. They are going to determine all of your features. Color of eyes, color of hair, your digestive system, all the things. Okay? Okay. Now, we are not going to talk about 3-7. So we're not talking about transcription. We're not talking about translation. We're also not going to talk about 3-8 with the exception that we are going to talk about mitosis and meiosis. And sometimes I say meiosis, sometimes I say meiosis. I mean them in the same way. I'll try to be consistent, is meiosis. But mitosis, meiosis. Remember, all cells come from pre-existing cells. So, we have to divide to make new cells. So, if I am dividing and just duplicating myself, I am going to use mitosis. And what I do is I take a camera, I take a picture of myself, I copy it, I move over, and I paste it. And I'm an exact replica. Boom, boom. Okay? That's mitosis. If I am dividing through meiosis, I am not going to be an exact replica, and I'm not going to have 23 pairs of chromosome when I'm all done dividing. So where would I not want 23 pairs of chromosome if I'm a cell and I'm dividing? Where would I most likely be reproductive? Right, yep, reproductive, yep. Reproductive system, when we're talking about the gametes, right, the cells that are going to help reproduce, right, I don't want my gametes to have 23 pairs of chromosome, because I'm going to have chromosome from mom, I'm going to have chromosome from dad. So I only want half. Okay? So meiosis, the end result is that I am not a duplicate. I'm not exactly the same. And I only have half the genetic code. So instead of 23 pair, I have 23 chromosome. Okay. Okay. Make sure that you got that one in your head. Make sure you got that straightened out. Okay. We'll see it again in development and inheritance down in chapter 20. Now, I imagine that you guys have heard of cancer and tumors. So, neoplasm is just a tumor, a mass of abnormal cells that are growing. And those abnormal cells that are growing can either be benign or they may be malignant. Benign is usually only problematic if it's in a place where it's constricting something or, something like that. Malignant is something that will replicate and travel and replicate somewhere else. So malignant. And that's called metastasize, to metastasize. Okay. Okay. I believe that covers chapter 3. Again, if there's any questions, feel free to jot them down. Shoot me an email. Don't do it alone. Let me help facilitate your learning. Let me get you some good resources and guide you through the process, okay? Save you some time, okay? All right, and we'll be on to the next chapter. Thank you.